So very good afternoon, everyone. I welcome you all on post-lunch session of the day third of first IBRO-APRC Associate School on advances in nanoneurotherapeutics and neurological disorders. We have with us our next speaker of the day from BITS Pilani, India, Dr. Deepak Chitkara. Dr. Deepak Chitkara is an associate professor at the Department of Pharmacy, Birla Institute of Technology and Sciences, Pilani. He obtained his B.Pharm degree from UIPS Punjab University, Chandigarh in 2004, followed by Masters in Pharmaceutics from NIPER, Mohali. Further, he obtained his PhD in Pharmaceutical Sciences from NIPER, Mohali. He was an exchange research scholar at the University of Tennessee Health Sciences Center, Memphis, for 1 year. After that, he did his postdoctoral training at University of Nebraska Medical Center, Omaha, in New York, in the area of mRNA therapeutics for pancreatic cancer in Professor Mahato's lab. His research interests include the nano-based delivery system for small molecules, miRNAs, and CRISPR-Cas genome editing tool. His lab is well funded from various government agencies, including DBT, DST, and ICMR, and also the pharmaceutical industries. He has published over 60 peer-reviewed publications, edited 1 book, and currently he is editing 2 special issues, one for Journal of Controlled Release on brain targeting and other on protein and peptide therapeutics for Journal of Pharmacology and Experimental Therapeutics. Further, he has filed 6 Indian patents and 1 international PCT patent application, out of which 4 Indian patent application has been granted. He has also the founding director of faculty lead nanotechnology-based startup Nanogrid Innovation Private Limited, incubated at Pilani Innovations and Enterprises Developmental Society at BITS Pilani, to further take up the commercialization activities. Today, on this occasion, he is going to deliver his talk on nanoneurotherapeutics for brain tumors, their various challenges, opportunities, and advancement. So we welcome you, sir.
Yeah. Thank you, Dr. Arti. So I'm audible?
Yes, sir, you are audible.
Okay, so just let me share the screen.
Sure, sir. It is visible, sir.
Okay. Yeah. Thanks for confirming, and thanks for giving the introduction. Rather, in fact, I mean, it's a good opportunity for me also for presenting the work and also the updates which are happening in this particular area of brain tumors. And with that, I would like to thank IBRO and APRC to conduct this school. And again, I've seen the list of the speakers, and they are really good ones. And I've gone through that lectures. They are really great. So it's a nice platform, and I would like to congratulate that to you guys. So again, I would like to thank the director, Dr. G.D.Gupta, the convener, Dr. Charan, and of course you, Dr. Arti, for this organizing this school. So basically, today I will be talking on the nanotherapeutics for the brain tumors. What are different challenges which are there? What are various opportunities and advancements that had happened in the field, and vis-a-vis what we are doing in our lab as well, in order to achieve the objective of increasing the lifespan in this particular disorder. So to start with, first I would like to just give an intro of what we are doing at our lab at BITS Pilani. So basically we are designing different carrier systems, which involves the polymeric systems as well as the lipidic systems along with their hybrids, to deliver small molecules, microRNA therapeutics, and CRISPR-Cas9 ribonucleoproteins for genome editing applications. So basically the diseases which we are targeting are the brain, where we are specifically looking into the glioblastoma, and apart from that, the ocular diseases, where we are looking into the retinal dystrophies, and on the topical side, on psoriasis. So in this way, varied areas, we are trying to deliver these small molecules or microRNAs, siRNAs, as well as designing different conjugates so that we can improve the delivery. So today's talk will be majorly towards the brain delivery of these small molecule conjugates, and particularly the temozolomide, which is one of the first line treatment for glioblastoma multiforme. So to begin with, if you see nanotechnology, it has advanced a lot. And first thing is the difference what we can see between the nanoscience and the nanotechnology. So basically, if you look at the history of it, so nanoscience where we are dealing with these structures and molecules to study those which are in the scale of nanometers, particularly in the range of 1 to 100 nanometers. And when we are applying for certain practical applications, say in case of medicine, we talk about the nanomedicine. There we have the term nanotechnology that comes into the picture. And if you go back, the nanotechnology, the concept or the hypothesis rather, was first discussed in 1959 by the renowned physicist and the Nobel Prize laureate Richard Feynman in his famous talk, which was titled as "There is a plenty of room at the bottom." So if you have seen that talk, it is also available on various medias and YouTube as well. So basically that hypothesis was put forward that is it possible that can we write 24 volumes of Encyclopedia Britannica on the head of a pen? And also another hypothesis or vision that was given that, can we use the machines to construct the smaller machines and down to the molecular level? So based upon that, some vision was created and people have started looking into this field of nanotechnology. And then he was also considered as the father of the modern nanotechnology because of the vision that he has provided. And in fact, after 14 years, the term nanotechnology was used by a Japanese scientist, Norio Taniguchi. And he basically defined this term that what comes under the nanotechnology. So basically, based upon that vision, in his famous talk, he also gave two challenges. The first challenge was, is it possible to miniaturize a page of text by 1/20,000 in a linear scale, so that it was readable with an electron microscope? And another challenge he was given to the scientist community, that is it possible to build a functioning electric motor within a 1/64-inch cube. So these two challenges he has given to the scientific community and asked them that if somebody solves these two challenges, $1,000 prize would be given to that person. And do you guys guess that for how long it would have taken to solve these challenges? So any thought on that? So if you see, basically within a year, the second challenge was solved, wherein William McLean, the Caltech graduate, has presented Feynman with a working motor far smaller than the pinhead. And you can see over here the Tom Newman used an electron beam to etch the opening of Bacon's Tale of Two Cities. It is a novel which was written on a 200 by 200 micron square plastic. So these two challenges were solved later, and again both of them won that $1,000 prize money. But what has been done with this, that the idea of going to the nanoscale has been envisioned, and people have started looking into this field, not only in the medicine area, but in all the areas, be it textiles, be and a lot of progress has been taken. Particularly if you look in the nanomedicine part, where we are looking into the advantages which this field can give. Now this term nano has been, again, you all can appreciate that this is now being used as a buzzword, that nanotechnology is being used, and we can see a lot of advantages that could also be achieved, though there are certain challenges that has also come up being in the nano size range. So there are a range of applications when we are bringing it down into the nanometric size range. So what are the positives and the negatives which are with this particular field? Say when we are talking about the nanomedicine. See, if you are converting a molecule, a drug into a nano size range, or loading it into a carrier system, which is in the nano size range, it has been very well now established that we can see higher efficacy of it. That results in a decreased dose. There is a possibility of achieving targeting to a particular site. And we can also tune the release based upon the systems, say PS sensitive systems could be designed, or enzyme sensitive systems could be designed, thermosensitive systems could be designed to control the release characteristics. Now, these all have been possible by converting the matter into the nano size range because of the unique properties that has come into it. But there are bottlenecks as well. And nowadays, if you can see that there are many systems that have progressed to the clinical trials and are also available in the market for various diseases. Like we can take an example of the liposomal formulations which are available, or the particulate formulations like we have Abrixane for paclitaxel albumin nanoparticles. But the journey was not easy. So there are many bottlenecks and challenges that the researchers have faced to make it feasible. Particularly, some of the bottlenecks that has been looked into is the scalability issue. Because when we are dealing with a matter at the nanometric size scale, the performing or designing these formulations at a lab scale in milligram or gram level is somewhat not much challenging, but when we are scaling it up, are we able to retain the same characteristics, same size distribution? So those are the challenges. And now there have been technologies which have been utilized to scale these things. Then another issue is with the reproducibility of the systems. So are we able to reproducibly manufacture the formulations at a much larger scale? Reason being, all the properties that we are getting, the benefits that we are getting, that is related to the size of the formulation, size and the size distribution in particular. Right? If that changes, there will be a change in the properties. And when we are dealing with large volumes at a much higher scale, there are chances. So it is always considered as a bottleneck to have the scalability with reproducibility. Another challenge with these formulations was with regard to the stability. Now, what happens that if we are going to a nanometric size range, there is an enormous increase in the surface area. Now, chances of the particles, coming close to each other, then interacting with each other and aggregating, that has also been increased a lot. And in order to stabilize that, various surfactants, combination of surfactants have been used, or other strategies have also been utilized. But definitely, stability is another major concern when we talk about the nanosystems. And the cost effectiveness. So if you can just go back and look at the price of a conventional formulation and the price tag which a nano formulation can have, you can find out the difference. Definitely, there is a lot of input that has been done by the companies, by the innovators to bring that formulation out, and that in the nano size range, and that will add to the cost of the formulation. But one of the challenge of bringing these formulations to the market is to make these systems also cost effective. Otherwise, they will be out of the reach of a common person. So these are some of the bottlenecks. Although there are several advantages these systems is given, but these bottlenecks also has to be looked into. Now, particularly when we talk about the nanotechnology and designing formulations, say if you are designing formulations for brain targeting, or to deliver these molecules to a particular region in the brain, say brain tumors, or specifically if we want to deliver to the cancer stem cell population, that too residing in the brain tumor, there are various facets we need to see. And that is from the scientist perspective. But apart from that, if we want a formulation to reach the market, we also need to consider other facets. So for example, the market potential, the technical and the scientific advancements. So basically, there are three components that we always have to look when we are designing such formulations. One is the market potential. If we are selecting a particular molecule to design or to develop a formulation, what is the market potential of that molecule? Whether there is any clinical need of it. So a clinical unmet medical need has to be clearly identified. And what are the technical and scientific advancements which are available or required to achieve the goal? So until unless these are not identified, the nanotechnology could not be used to its fullest. Now, being from the pharma background or the medical background, our major focus lies on the clinical need, to identify the clinical need. And when we talk about the clinical need, we can broadly classify it in terms of identifying the safety, efficacy, or compliance. So these are the three pillars I can say And if you are dealing with a molecule, say particularly for tumors, able to improve the safety profile of the molecules which are already existent by converting it into a nano form, or is it possible to improve the efficacy also by delivering them to the target site in a better way, or at least can be able to improve the patient compliance so that he can take the medication without any break. So if we were able to achieve any one of these, then definitely there is always a scope for that technology to go to the market. And this nanotechnology has enabled delivering of several difficult to deliver molecules that we will see later on, that how these have been used, what advantages they have given. And you may see over there that either of these, or rather most of the things are achieved. Say that the formulations, they will be having a better safety profile with a better efficacy as well. But even one of these is being achieved, then that formulation is definitely good and could be taken forward. So there is another approach that normally being used when we design these different formulations and to integrate the whole you can say the project. So it is called as a 4D approach, where we have the disease, drug, destination, and delivery. So if you look at the disease right now, if you're talking about the glioblastoma multiforme, what are the challenges with that particular disorder? How we can make our drug to reach that particular site? So the location of the disease, the barriers which are involved, those are very important in identifying the right delivery system. But that's not all. We also have to look at the drug molecule. Now, molecule could be either a small molecule, it could be either a protein or a peptide or a gene therapeutic agent, say a siRNA or a microRNA. Now, that has to be basically delivered to the site, which is glioblastoma multiforme, the tumor cells. And in order to achieve that, all the physicochemical properties, the biopharmaceutical properties of that molecule has to be assessed properly and integrated with the delivery system. And here, if we talk about the delivery system, particularly focusing on the nanotechnology-based systems, so there are a variety of carrier systems which are available. If we talk about, we have the nanoparticles which are lipidic in nature, or polymeric systems, or their hybrid systems, the dendrimers, polymeric micelles, and so on. So which of these delivery systems has a capability to deliver the therapeutic agent of interest? So that has to be integrated together so that we'll be able to deliver the molecule to the right destination, so that we'll be able to achieve the maximum therapeutic benefit. And when we talk about the destination, again, it's not only that where we want the therapeutic agent to reach, but it also matters that which pathway it is being followed or which route is being used. Say when we talk about the brain delivery, there are formulations which are given via oral route. The drug absorbs and goes into the systemic circulation, then crosses the blood-brain barrier and reaches the brain. Then there are formulations also which are injected. And also another route which is nose to brain delivery that has also been explored. So depending upon the destination, the route which we are using and where we want our therapeutic agent to reach, the delivery system could be looked into and accordingly modified. Now coming on to the gliomas or the brain cancers. So as per WHO, these gliomas has been classified into 4 different grades depending upon the severity, how these tumors are growing, what is the likeliness of the spread, what are the chances of the relapse. So if we talk about the grade 1 is also called as juvenile pilocytic astrocytoma. So here the tumors are slow growing and they are unlikely to spread and less likely to reoccur, and often a surgery could be used and this could be cured by the surgery itself. Then the next grade is the grade 2, which is also a astrocytoma, where the hypercellularity is increased, though there is no mitosis, no vascular proliferation, and no necrosis. In this case, the tumors are also less likely to grow and spread, but the relapse chances are there. They are more likely to come back after the treatment. Then we have grade 3, which is anaplastic astrocytoma, where the hypercellularity rate is increased, there is a high rate of mitosis, though no vascular proliferation is there and no necrosis. Now, these tumors are also more likely to have rapidly dividing cells, but since necrosis is not there, they are not dead cells, but they can grow quickly. And finally, the grade 4, which is the glioblastoma, which is a more severe form of the brain cancer. Here, the hypercellularity is very high. The mitosis is also very high. There is a presence of vascular proliferation, presence of necrosis, and the cells are also actively dividing. There has been a growth, the abnormal blood vessel growth, and there are also the areas of dead tissues. So the chances of survival in grade 4 are very less. So if you look at the GBM, so it is one of the rather most prevalent tumor and deadly as well. So it is malignant, highly aggressive, and as we have seen that it is from the astrocytic lineage. Now, what are the challenges for the treatment for glioblastoma multiforme? So if you overall look at the location, the heterogeneity, and the diffuse and infiltrative growth of this the disease, that makes it highly challenging to cure. Now, since it is located in the brain, so any molecule which we are giving, it has to cross the blood-brain barrier and goes to the brain tissue, and after that it has to reach the tumor compartment. And in that specifically, it has to be taken up by the cells to show its activity. So much barriers it has to cross. Plus, at the same time, there is tumor heterogeneity as well. Like there is a presence of the cancer stem cells that can ultimately lead to the relapse. So how to deal with that? So those are some of the challenges which are there with this particular brain tumor. Now, this has come because of the various mutations that had happened. Like there is a loss of heterozygosity on the chromosome arm 10q. And it has been seen that it is associated with a poor survival and also contributed to the development of GBM. Then there is also a p53 deletion. Now, this is involved in the tumor suppression, right? This p53 deletion resulted in the lack of tumor suppression. Then there is upregulation of EGFR, VEGF, and also MDM2 amplification had happened. Then MGMT amplification is also been there. Now, MGMT has a role in the DNA repair, right? And it is once it is upregulated, so it is known to have influence on the chemo resistance, particularly towards the temozolomide, which is one of the first line treatment for glioblastoma multiforme. And if you look at various factors that have resulted into GBM that have contributed, if we look at the risk factors, there are many like obesity, smoking, alcohol that has been related to it, the phone usage, radiation, exposure to the radiations, plus age. As we are growing, there are higher chances of the glioblastoma. Then again, if you look at the gender, the males are more prone towards the GBM. Then hormones like progesterone, it has a role in protecting the glioblastoma occurrence. Then genetics is also involved, where there are several mutations, particularly ATRX mutations, TP53 mutations, and so on, which are involved. And use of certain medications, use of cell phone, exposure to heavy metals have increased the incidence. Now, if you look at the incidence, it is around 3.19 to 4.17 per 100,000 person. So the incidence is also quite high over there. Now, what are the treatment approaches? Now, we have seen that glioblastoma multiforme is one of the deadly form of the brain cancer, which is having a location which is difficult to access. Plus, there are other issues as well for the treatment. So, what is normally done? Now the modality that has been followed is first we have the surgical resection, which is normally the first step if the tumor can be safely resected. Now, once the tumor is resected, then there are BCNU or carmustine wafers which are available that could be placed in the tumor cavity that can provide a localized chemotherapy. Now, along with that, there is a KPS status, the Karnofsky performance status is measured for the patients. Now, if that status, depending upon the status, either the chemotherapy or the radiotherapy or a combination of these two is started. So in this case, temozolomide is used as one of the first line treatment for the tumors for this glioblastoma multiforme, either alone or in combination with the radiotherapy. Now, if you look at the timeline for the development of this molecule, so this molecule was discovered somewhere in 1992, and it was developed. Its mechanism of action was also defined. Then it went into the phase trials. The first clinical phase trial was done in 1992, and then it was licensed out, and it has been approved by the FDA as the second line treatment initially in 1999. And later on in March 2005, US FDA has approved temozolomide along with radiotherapy for treatment of glioblastoma multiforme in the newly diagnosed adult patients. Then in 2008, the total overall sales, if you see over here, are 1.02 billion, which is quite huge. So it is one of the blockbuster drug molecule which is being used. And then later on, the intravenous formulation. Now, earlier this was for the oral formulation was available. The intravenous formulation has also been developed, and it was approved in 2009. Now, this is with regard to the molecule that is being used for the treatment of glioblastoma multiforme and its journey. But alongside, there are also challenges that has come up with this molecule. One of the challenge is with regard to the resistance. So MGMT is an enzyme which is involved. So MGMT is basically a methylguanine DNA methyltransferase. Now, the upregulation of this enzyme has resulted in the development of resistance towards temozolomide, and later on we'll see that how it is causing that resistance. So in order to avoid that, various methods have been used, either by using nanotechnology based approaches to deliver the molecule more effectively, and also using some MGMT inhibitors along with it to improve the efficiency, or using combination chemotherapy, say utilizing siRNA or microRNAs or other chemotherapeutic agents like doxorubicin. So those strategies have been utilized to overcome the resistance and also improve the efficacy of temozolomide. Now, what is this temozolomide molecule? So if you can see the structure over here, so this is temozolomide. It is basically imidazole tetrazine derivative, and it is a prodrug. So it releases this methyl diazonium ion that goes and basically methylate the guanine residues O6 and N7 of the guanine residue on the DNA, resulting in the autophagy mediated cell death. So it is used as a first line treatment, available in the form of capsules, tablets, as well as injections. Now, this molecule, when given orally, is 100% bioavailable. Then you may ask the question that if it is 100% bioavailable, then what is the problem of designing new formulations? Now, though it is 100% bioavailable, this conversion of temozolomide to its inactive metabolite AIC and MTIC, resulting in the release of this methyl diazonium ion, happens at pH 7.4. So though it is stable in the acidic pH, but as soon as it comes in the pH of 7.4, its metabolism starts. Now, the amount of drug that is reaching the brain, this temozolomide is limited. It's hardly 10% that reaches the brain, and particularly the brain tumor, the fraction is even smaller. So that is why the doses are quite high for this molecule. So if you can see the fate of temozolomide after the absorption, that what happens that this temozolomide, once it reaches the plasma circulation in the blood, in the systemic circulation, though 100% bioavailability is there, it is having a half-life of only 1.83 hours and is converted to MTIC. So MTIC, which is the intermediate form, has a half-life of only 2 minutes, immediately forms the AIC and the methyl diazonium ion. So if it is converted in the systemic circulation, then this methyl diazonium ion may not be able to cross the blood-brain barrier. So it is a temozolomide intact that crosses the blood-brain barrier, and only 10 to 20% of the intact TMZ reaches the brain. So once it goes to the brain, it has to go to the tumor cells where it is uptaken. And this again, the metabolism happens inside the cell, resulting in this methyl diazoniamide that can go and intercalate with the DNA, methylating the guanine residues of the DNA. So this will lead to the autophagic death of the cells. But how much percent of actually reaching the site and causing and is being utilized for the efficacy is very limited. So that is why there is a definite requirement of designing new delivery systems. Plus, at the same time, there are there are high chances of development of resistance by various mechanisms. Like there are epigenetic mechanisms which are also involved, that involves the hypermethylation of the O6 MGMT promoter region. Now there are efflux systems which involve the cellular mechanisms, then tumor microenvironment which is hypoxic, and the presence of the cap junctions that also limits and contributes to the TMZ resistance. And there is a direct mechanism that involves the MGMT enzyme. So the role of this enzyme is to demethylate the guanine residues. Now we have seen that the methyl diazoniamide that is released from temozolomide is basically methylating the O6 and N7 of guanine residues. And this enzyme demethylates that. So if there is an upregulation of this enzyme, then it can reverse the action of temozolomide, resulting in the development of chemoresistance or temozolomide resistance. So these are the challenges which has to be solved. So if we see temozolomide, the limitations which are associated with it, it is having a short half-life, there's a rapid metabolism which is happening, and it starts happening as soon as it enters the plasma circulation. Only 10 to 20% of the administered dose reaches the brain, plus there is a development of resistance. So definitely there is an unmet medical need that necessitates the use of these new technologies and novel strategies for TMZ delivery. So basically some part of that we have already discussed in one of our previous publications, where the new strategies have been discussed, what kind of modifications that could be done for temozolomide delivery. Now, if you look at the barriers that can affect the delivery of any therapeutic agent. So here we are talking about the temozolomide. Now it has to be converted into some suitable carrier system that can deliver it to the site. Now, one thing is that it could be taken orally, which is one of the formulations of temozolomide which is available, and another one is via intravenous route. But whatever way we give, it has to reach the systemic circulation. And once the carrier system is in the systemic circulation, then we all know that the REA system that can clear off the carriers, because opsonization happens and these carriers are taken up by the macrophages and they will be cleared by the REA system. Plus, there are chances of degradation, instability that also happens depending upon the type of molecule or the carrier system which we are dealing with. But once it's sustained in this, it has to cross the blood-brain barrier. Now, when we talk about the blood-brain barrier, and we'll see in the next slide the more details of it, that what blood-brain barrier constitutes. Now, this carrier system has to cross the blood-brain barrier, which is having tight junctions, and there are presence of the pericytes and astrocytes. So this blood-brain barrier is basically controlling the complete homeostasis of the brain, and it is difficult to breach for these carrier systems. But when the tumors are growing, particularly we have seen that in case of grade 4 tumors, this glioblastoma multiforme, there is hypervascularity, hypercellularity has happened over there, and there is high vascularization. So the blood-brain barrier changes. So we have now the blood-brain tumor barrier. Now, apart from that, in these tumor cells, if we specifically focus on these tumor cells, there is also a subpopulation of cells called as the cancer stem cells, which has a potential for self-renewal and also differentiation. Now, they are the one which are chemoresistance, the radiation resistance, and can have the high tumorigenicity. So the drug molecule what we are using can kill the tumor cells, but there is always a residual stem cell population that remain and ultimately lead to the tumor relapse. So if you look at this overall barriers, and if you want to design the system, we have to take care that the carrier system could take the drug across all these barriers, reach the site and deliver the therapeutic agent. Plus, there should be a capability that if we are doing a combination treatment, they should be able to kill these cancer stem cells also, so that the tumor relapse could be avoided. Now, what is this blood-brain barrier? So basically, this blood-brain barrier is kind of a single neovascular unit that is composed of the cellular components and the extracellular components. So we have the endothelial cells. So these endothelial cells are basically covered by the pericytes. Then we have the astrocytes. Along with that, we have the extracellular matrix. So apart from that, there are certain other proteins which are forming the tight junctions. So we have these claudins, occludin, junction adhesion, and cytoplasmic associative proteins that basically forms a very dense network or a very dense barrier around the blood capillaries that are going to the brain. So this lead to high paracellular resistance towards the pathogens or any injury or any molecules or proteins, and the whole process could be controlled. So, and is very important in maintaining the homeostasis. Now, if you see in case of temozolomide, there are challenges that we already know now, that this is a molecule where there are problems with regard to the stability of the drug, that it degrades at the physiological pH, resulting into MTIC. And also there are chances of chemo resistance that happen because by various mechanisms. Now there are several reports where nano-based systems have been utilized to deliver this molecule, like liposomes have been developed, solid lipid nanoparticles, dendrimers, the nanoparticles. So they have been used to deliver this molecule. But again, delivering this molecule is not that easy. Reason being, there are problems related to the physicochemical properties of this molecule as well. So when we are loading this into a nanocarrier system, one of the important things that we need is that we should load efficiently. There should be high efficiency. But at the same time, high drug loadings are required since the dose of the molecule is high. Now, if you look at the water solubility, the drug is having a water solubility of 5 mg per ml. So because of this high water solubility, it is always difficult to load this molecule inside in a nanocarrier system. Though there have been several reports where modifications has been done in the process, or those processes has been optimized, wherein high encapsulations have been reported, like certain nanocomplexes have been prepared, and nanostructured lipid carriers have been prepared that have shown 83% encapsulation efficiency. Then SLNs are there, right? Nano cages, liposomes have shown around 35% in rapid efficiency. Then DPPC liposomes are there that have shown 87% encapsulation efficiency, and so on. So systems are there, no doubt, but this is always challenging when we are loading a water-soluble molecule into any nanocarrier system. So one challenge is loading it inside with high encapsulation efficiency, and another one is controlling the release of the molecule from that system, because it is always tend to come out at a faster rate. Now, if you look at the outcomes are quite uniform. If we see that there is always an improvement in the anti-tumor activity in vitro and or in vivo studies. Plus, these literatures have also reported that there has been a significant reduction in the toxicity. So this is also obvious that when we are using any nanocarrier system, we are doing it for these advantages, and definitely these systems are capable of providing these advantages. Now, another type of strategy that has been used, since this is a hydrophilic molecule which is having water solubility, appreciable water solubility, which makes it difficult to load into carrier systems. So another strategy that has been utilized is to conjugate it, conjugating it with a polymer system or with small molecules. Like there are polymers which have in their backbone the carboxylic acid group. Now, temozolomide could be slightly modified, and then conjugation chemistry could be made in such a way that using a linker, these polymers could be conjugated. Then there are several small molecules also with hydroxyl or carboxylate groups, which are the reactive sites, where it has been conjugated. Now, these conjugates again have shown an improvement in the stability of the temozolomide, provided a sustained release because it takes time for the bond to break. Then there is also an improvement in the circulation time. The blood-brain barrier permeability has also been shown to increase, plus targeting has been achieved, and definitely the improvement in the cellular uptake for these conjugates. So these small molecule conjugates or polymeric conjugates, so there are several examples of these which are there in the literature, have provided advantages, plus they are also associated with certain limitations. So when we look at the advantages of small molecule conjugates, now these conjugates, if we control the ratio of the hydrophobicity and the hydrophilicity in that system, these conjugates may be made self-assembly. Plus they could be conjugated, like for example, doxorubicin has been conjugated with temozolomide. So a synergistic activity could be achieved. Plus the small molecule conjugates have been made with parallel alcohol and also with fatty acids, where synergism has been seen. Plus also the temozolomide stability that has been improved. On the other side, if you look at the conjugates, when we are designing, they are always considered as a new chemical entity. And it has to pass through all the trials from safety to efficacy. And again, when we are conjugating with a small molecule, what is the ratio with which we are conjugating? Because the number of groups will define that. Now, normally it is always a 1-to-1 ratio. Whether this ratio is synergistic or not, or it may be effective or not, that is always a questionable. On the polymer side, again, the self-assembling systems have been generated. A higher drug loading have been achieved where multiple groups, if present on the polymer backbone, they could be conjugated with the temozolomide to improve or to have higher drug loadings. But challenges are also there with regard to the toxicity of the polymers, plus controlling the conjugation efficiency from batch to batch, which is always difficult to control. So these are some of the challenges which are there with the polymers when this drug molecule has been conjugated. So this part of this, what are those polymer conjugates or small molecule conjugates for temozolomide are there. We have recently covered in one of our article in Journal of Controlled Release. Then coming on to the polymer drug conjugates of temozolomide. Now initially this paper, Patil et al., so basically they have conjugated the temozolomide with polybeta-L malic acid. So polybeta-L malic acid is having the carboxylic acid backbone. So temozolomide definitely has to be modified accordingly to conjugate it onto the polymer backbone. And there it has been seen that improvement in the cell viability, right? The toxicity which is there when these conjugates have been used. In both cell lines, they have tested in U87Mg cell lines and T98G cell lines that the conjugates have shown. And these conjugates have also shown good drug loading, like you can see over here, around 17% of the drug has been conjugated with the polymer. Plus conjugates have been also prepared with other polymers, like over here we have this poly-2-ethyl 2-oxazolane, PET or ZTMG conjugates. Now here also these conjugates, basically these self-assemble, and around 4% of loadings have been achieved with these conjugates. And you can see in the data here that these conjugates were able to have better efficacy when tested in cell lines and also in vivo animal models where an improved lifespan is obtained. Plus, it has been checked that the efficacy of the drug interacting with the DNA, they have also been tested by using the H2AX method, so immunofluorescence of this. So they have tested that it is also intercalating with the DNA and interacting with that. So after conjugating, the efficacy is not changed. So that has also been tested and is proven. Now, apart from the polymers, as I said, the small molecules have been also explored. And in case of small molecules, the fatty acids and fatty alcohols have been explored for conjugation. And one such molecule is Neo-212, which is a temozolomide conjugate of where temozolomide was conjugated to parallel alcohol. Now, this conjugate has also shown a good efficacy in various cell lines, in resistant cell lines as well. As you can see over here, then U251TR, then LN229TR2, these cell lines, they are resistant to temozolomide. But this Neo-212 is efficacious, have shown the cytotoxicity in these cell lines as well. Plus, there are, other fatty acids have been used, and in our lab, we have used various fatty acids for conjugating small molecules. Now, those have also been used for conjugating with temozolomide. So, if you are conjugating any fatty acid molecule with temozolomide, it gives several advantages. One thing is that it converts it into a hydrophobic, or imparts some hydrophobicity into it. And if that is properly calibrated, the system could be made self-assembling also. It could, since there is an increase in the hydrophobicity, it could be loaded into a carrier system. If we are loading in some nanoparticulate system, that could be achieved. Plus, fatty acids, they have already been shown to have synergistic action, to have apoptosis, to cause apoptosis. So those advantages could also be achieved from these fatty acids. Now, at the same time, fatty acids have also been shown to increase the blood-brain barrier permeability. So using a proper conjugation chemistry, the conjugates of the small molecule could be made with the drug, so that these properties could be imparted into it. So basically, there are several opportunities with these fatty acid conjugates when we are conjugating it with therapeutic agents. Now, several reports are there, like for example, oleic acid has been shown to induce apoptosis and autophagy in squamous cell carcinomas. And there are several anti-cancer molecules like doxorubicin or paclitaxel that has been conjugated to fatty acids. Like doxorubicin is conjugated to oleic acid and then loaded into nanostructured lipid carriers. And linoleic acid has been conjugated to paclitaxel and has been shown for its therapeutic efficacy in glioblastoma. So there are various studies where these conjugates have been explored. Now, if we talk about the small molecule conjugates of temozolomide, now temozolomide is having a CONH2 group over here. Now, if you are conjugating it with a fatty acid, so there should be a proper chemistry which is involved. We need the proper functional groups that can react with each other to form the linker or the conjugate with each other. Now, CONH2 group is normally converted into a acid group to form a temozolomide acid. And on the other side, what we have done that we have converted the fatty acid into its hydrazine derivative. So N group is modified from carboxylic acid to NHNH2. And now the acid group could be conjugated with the NH2 group to form the temozolomide fatty acid conjugate. So basically in our lab also, we have screened various conjugates of temozolomide, wherein we have conjugated temozolomide with oleic acid, with palmitic acid, with linoleic acid, and tested for their efficacy in cell lines and in animal models. Now the data here, you can see that these conjugates were indeed quite effective when they are being, if we compare it with the temozolomide in both the cell lines, wherein we have used the C6 cell line and U87Mg63 cell line. So both these are showing the better efficacy for the conjugates. Plus, when these conjugates were tested for the apoptosis, so there also we have found that these conjugates were able to induce the apoptosis in both the cell lines. So this is for the C6 cell lines, where temozolomide alone has shown around 18% apoptosis, including both early and late apoptosis. While if you are talking about the conjugates, say temozolomide oleic acid conjugates, TOA stands for the oleic acid conjugate, it is showing 81% apoptosis. Then with palmitic acid conjugate, it is around 82% apoptosis. So there is a definite improvement in the apoptotic potential of these conjugates. Plus, if we further look into the effect on the MGMT. So in this particular cell line C6, once they are being treated with temozolomide, there is an increase in the MGMT. So which is also known for the chemo resistance. But these conjugates did not affect the MGMT status of the cells, indicating that they are not having any potential to cause this temozolomide resistance. So similar results we have seen in case of another cell line, which is U87Mg cell line, where also we have observed an improvement in the apoptosis when we have used this temozolomide or the conjugates. And also in case of the MGMT expression, similar observations were seen for U87Mg cell line. Now, having a cue from these data, we have taken one of the conjugate, which is temozolomide oleic acid conjugate for further studies in glioblastoma multiforme tumor in rats. So basically, C6 cell lines were used to develop an orthotopic glioma model, wherein these C6 cells were injected into the at a particular location. So it is 2 mm lateral, 2 mm posterior, and 5 mm down to the bregma region in the Sprague Dolly rats. And further, these animals were evaluated for their physical symptoms, as well as the tumors were later on taken out and they were examined for this development of these tumors. And we can clearly see over here that in case of a normal saline injected rat, both the right hemisphere and the left hemisphere, the widths are same. While we can clearly see the tumor growth in case of the C6 cell injected rats. Now, this model was validated and further used for the analysis of the temozolomide oleic acid conjugate for its efficacy. And what have been observed that in case of this conjugate, when we are comparing, there is a definite improvement in terms of the tumor growth. So that we can see once we sacrifice the animals, we can see for the ratio of the right and the left hemisphere, where it has been observed that there is a decrease in the TOA treated conjugate treated group. Plus, we can also see in the survival of the animals that has been prolonged a lot as compared to the positive control. As compared to the positive control, we have given, and so this is the green is for the temozolomide, where we have not seen any death up to 80 days. While in case of temozolomide, there have been animal deaths that had happened at different time points, and marked improvement has been seen in case of the survival of the animals, indicating that these conjugates were indeed quite effective. Then in the literature, if we see, so these are with respect to the conjugates that we have done, but we have also looked into the literature what others are doing in terms of designing the different types of conjugates or small molecule conjugates for this. So there have been reports also where people have tried conjugating temozolomide to doxorubicin, as in case of Liu et al. They have published this in ACS Applied Materials and Interfaces, where temozolomide was conjugated to carboxylic acid and has been tested. So there again, the results were promising in terms of improving in having more cytotoxicity in cell line models. Then there are other reports also. Now, what are the advancements now that had happened in this particular field? If you look that we started with the treatment of glioblastoma multiforme, where temozolomide was a drug that has been approved first as a second line treatment, later on as a first line treatment along with radiotherapy. But there are also challenges which are associated with this molecule. And in order to overcome those challenges, various formulations has been tried from both the facets. One is using the nanocarrier systems to load that molecule physically and deliver that. The efficacy has also been seen in those formulations. Plus, using a conjugation approach where the drug is conjugated either to the small molecule or to a polymer. Apart from that, there have been other advancements which has taken place in the field, wherein the conjugate, this temozolomide, has been delivered along with the RNAi therapeutics, say using a siRNA or a microRNA for glioblastoma treatment. Plus, there have been advancements that have been taken place where certain antibodies or peptides have been used to cross a blood-brain barrier, or surface modification could be done. For example, utilizing the RGD peptide or transferrin or ApoE peptide. Then from the delivery perspective or from the route perspective, nose to brain delivery has also been explored for these therapeutic agents, so that we can directly reach the CSF, and from there it can go to the tumor compartment to show its efficacy. So basically this part we are right now covering in one of the special issue that we are doing in Journal of Control release, which is entitled at Drug and Nucleic Acid Delivery for Targeting to the Brain. Then there is another special issue so basically this first issue of JCR, which I am co-editing with Professor Mahato. He's at the University of Nebraska Medical Center. For herein we are dealing with all the strategies that could be utilized for brain targeting of the therapeutic agents. Then another special issue I've just started with Professor Anupma Mittal, which is on the protein and peptide therapeutics for the Journal of Pharmacology and Experimental Therapeutics. Again, the scope is wider over here, that and targeting specifically for the proteins and peptides delivery. Now, if we look at the RNA therapeutics that have been used for the glioblastoma multiforme, so here in both siRNAs as well as microRNAs has been explored for improving the efficacy of temozolomide in combination with that, or alone also they have been tested, because we have seen that there are several mechanisms which are upregulated or downregulated. Can we target those by targeting the mRNA and using RNAi? But utilizing these gene therapeutics is also not easy. Over there also different challenges come up. Since these gene-based therapeutics, they are highly negative in charge, plus high molecular weight and hydrophilic. So what strategies could be used to deliver those? Because when we deliver those in the naked form, they are bound to degrade. RNAs are there that can degrade these, and they may not be able to even cross the biological membranes or enter the cell. So various strategies have been utilized, using certain cationic lipids to form the lipid nanoparticles, or using nanogels or aptamer-based strategies. Certain lipoplexes wherein cationic phospholipids have been used, and they have been complexed with that. They have also been used for delivering. And also certain polymers, particularly the cationic polymers that have been used. And in our lab, we are majorly focusing on using these micelle plexes or cationic polymers. So we are designing various cationic polymers which is having the polycarbonate backbone, and onto which the different ligands, including the cationic chain, the morpholino group, or the cholesterol, they have been attached to improve the delivery of these microRNA therapeutics. Now, there have been reports where these therapeutics have been used. Say, for example, in case of the study by So et al., so they have designed the aptamer-like peptide functionalized liposomes for siRNA delivery for glioblastoma treatment. So herein, again, a marked improvement has been seen, and the target, which is basically cyclophilin, has been silenced by delivering the siRNA. The siRNA is basically EDB targeting. So herein, these results, both in vitro and in vivo results, have shown that it is going to the tumor when they are delivering it using these nanoparticles, and the effect is also clearly visible. Then microRNAs, they have also been used for glioblastoma treatment, like this anti-MER21 and anti-MER10B have been co-delivered by Ananta et al. Using PLG nanoparticles for the improvement in glioblastoma multiforme. And these two microRNAs are involved in the cell proliferation, development of chemoresistance, by affecting the expression of PDC, CD4 and P10 and HOX D10. So these anti-MERs for these two microRNAs have been delivered, wherein the positive effects of delivering it along with this temozolomide has been seen that there is an improvement in the therapy. Then, apart from these therapeutic agents that have been explored, say we have the temozolomide, then the siRNA or the microRNA delivery systems, or using combination with another drug molecules. Studies have also been done to deliver the drug from nose to brain, using nose to brain delivery for these nanotherapeutics. And there are quite a few examples wherein these therapeutic agents have been delivered. And one such good report is on the nose to brain delivery of temozolomide loaded PLG nanoparticles. So these PLG nanoparticles have been prepared by Chu et al., wherein these particles have shown a size ranging from 125 nanometers to 145 nanometers, and have been delivered through the nose, and a definite effect in the treatment, in the improvement in the survival has been seen when these nanoparticles were delivered in the animal models, in the glioma bearing rats. So a lot of advancements have been taken place in delivering this temozolomide and then combining it with different therapeutic agents. So now coming on to the summary, so basically we have seen that in case of glioblastoma multiforme, which is one of the most prevalent and aggressive brain tumor, so over there temozolomide is being used as a first line therapy that has been approved a way back. But there are several limitations which are associated with this molecule in terms of its stability, its degradation in the plasma, plus the development of chemoresistance. And for that, several nano-based approaches have been utilized for delivering it using different nanocarriers, be it solid lipid nanocarriers or a NLC-based system. Then another approach wherein the molecule has been conjugated to the small molecule or polymers have also been explored, wherein an improvement in the therapy has been seen. It has also been combined with other therapeutic agents such as siRNA or microRNA so that the pathways could be targeted which are involved in the resistance particularly. And another strategy that has been explored is delivering the drug through nose to brain to reach the CNS and this also provides great opportunity. So with this, I would like to thank everyone and for your patience listening. And also I take this opportunity to acknowledge the people who are involved in the on this temozolomide project that we are running in our lab. So basically we are working on delivering this temozolomide molecule using conjugates with a small molecule as well as with the polymers, where we are designing these polycarbonate based systems for delivering this. So Reena, Prabjit and Deepak Sahel, who are PhD scholars working with me on this particular project. And then we have a set of collaborators who are involved in various aspects of this project. And definitely the financial support from the ICMR as well as TBT for these temozolomide related work. So with this, I would like to thank everyone, and I'm open for any questions.
So thank you very much, sir, for excellent talk. Now the session is open for discussion. Yes, Simran, you can ask question. Simran ji.
Okay. Good afternoon, sir.
You can switch on your camera also.
Okay. Good afternoon, sir. Thank you for such interactive session. So, sir, my question is that what are the possible gaps between the preclinical to clinical when we talk about nanoneurotherapeutics in terms of cancer, especially for the cancer?
Okay, preclinical to clinical.
Yes, sir.
Right. One thing what I can directly see is in terms of the models that we have. Right. Are they actually recapitulate the same thing what we can see in when we are going for the clinical studies? Now, in case of clinical studies for cancer, definitely the subjects over there are the patients, actual patient population, and first safety has to be established in them. Now, when we are developing these nano systems, so how we are processing, we are first doing it in the animal models for the safety assessment and then for the efficacy assessment in these animal models. Now, here lies a big gap in terms of the efficacy, the models which we are dealing with. Say for example, glioblastoma particularly, say if you are doing it in a rat model using a particular cell line, whether it actually recapitulates the tumor that is actually existent in the patient population, number one. Number two, the mutations. Again, they are very difficult to recapitulate everything in terms of mutations. So this is with regard to the heterogeneity which is there with that tumor that varies in the preclinical and the clinical side. Apart from that, when we specifically look into the nano, in case of nano, another challenges do come up in terms of its biodistribution. What we see in case of animal models, are we going to see the same in case of humans? That is another big difference that will come up. And how these are going to show its effects. So say how much component of these nanocarriers are actually reaching the site to deliver the therapeutic. So this is from the efficacy perspective. Now there are issues also with the toxicity of these nanosystems. And in fact, another separate set of studies required to understand the nanotoxicology of these formulations, because there are few examples where the toxicities are not there with the conventional formulation, but it has been seen when the nano formulations are used. As in case of Doxil, if you know about the hand and foot syndrome, which is there with the Doxil formulation, a liposomal doxorubicin, but it's not there with the conventional formulations. So these are some of the gaps that needs a lot of addressal. And again, I mean, it is always difficult to fill in that gap as well. That is in fact an unmet need to recapitulate the entire preclinical setup and correlate with the human studies. I hope I was able to answer you.
Yes, sir. Thank you, sir.
Yeah.
Yeah, thank you so much, Dr. Deepak.
Yeah.
So thank you so much, Deepak, sir, for sharing this excellent talk and for sharing that much information about this drug on glioma. So I think I am the only non-pharmacist person here. So I'm just trying to understand the things going behind this chemistry and all these things. So my first question is that you have mentioned this drug that is TMZ is not able to treat efficiently just because of its half-life, because the half-life of just is very low. So I'm just wondering what is the time it requires to reach the brain once it gets metabolized to the system, like not metabolized, I would say absorbed in the system, because it's 100% available.
Yeah. So it's not with respect to the time, because the circulation, once it reaches the systemic circulation, then it is moving. These are dynamic processes which are happening, right? So if you see at the half-life of the molecule is temozolomide is having a half-life of 1.8 hours. So that means that in that 1.8 hours, its concentration reduces to its half. So even if it is 100% bioavailable, its degradation immediately starts in the plasma. So now we are having the temozolomide and its inactive metabolites, or the diazonium ion which is formed. It is only the temozolomide which is remaining can permeate through the blood-brain barrier, because there is slight hydrophobicity and that is required to permeate the BBB. Right, so it is only temozolomide that can go, but not the inactive metabolite which is happening, or the diazonium ion, which is actually shows the effect. So it has been
so
seen
so basically, what are the transporters which actually transport these like metabolites of this drug? Because I think the active component of this drug is the final metabolite, like which has even the shorter half-life than the TMZ drug.
Yeah. So basically, it is the temozolomide that can only permeate by diffusion. So it is not the diazonium ion that can permeate. So that is one of the problem, because once it is getting metabolized in the plasma, it stays, the diazonium ion stays over there. It cannot go to the brain. It is only temozolomide that, if it is intact, it can go to the brain, enter the brain tissue, the goes to the cell, the cancer cells, and over there, if it is converting into diazonium ion, that is showing its activity.
So don't you think, I think 1 hour, 1.8 hours are more than enough to utilize this drug by the brain?
I don't think so. That's what is there in the literature also. Because even if with this molecule, although it's a first-line treatment, but still the survival is not that much. It's having good activity. If you see in vitro cell cultures also, it shows good activity. But that is not translated in the actual tumors, when the whole biological milieu is there. So because of the challenges which is having. So one side is on the stability, that only 10% is going. But there are other issues which are more grave issues which are associated with this molecule, is because of this resistance, this temozolomide resistance that comes in. That may be related to some part with the temozolomide, its mechanism of action, because it is methylating the guanine residues at O7, at N7 and O6. And there is an increase in the MGMT enzyme that can demethylate. So that is one way it is getting this resistance. So that is why people are trying to overcome this temozolomide resistance by using other chemotherapeutic agents along with it, or using some strategies where gene-based therapeutics like siRNAs or microRNAs are being used. So those strategies are being also tried, because that is particularly related to the molecule mechanism.
Okay, so I have one more last question. So that might be a silly one, I would say. So let's say you just like encapsulate your drug inside this nanocarrier, then you just throw all the 100% to the brain, right? Okay. If the brain just utilize about 10 to 20 to 30% of the drug, then what would happen to the rest of the 70 or 80%? How it would get like metabolized, and how it would get back to the blood and get like excrete out from the system by the liver system and all.
So basically, as I've said, it is a complete dynamic system that is happening. So when we talk about the pharmacokinetics of a drug molecule, it's not a static part, right? It is getting absorbed, getting distributed, getting excreted and eliminated. So all these ADME processes are happening in general. But when we talk about this temozolomide molecule, temozolomide is also getting degraded, right? It can, it is stable in the acidic condition, but in the biological fluid, when the pH is 7.4, it is being converted to MTIC and then AIC. AIC is the inactive metabolite and diazoniamine is formed. So diazoniamine, this is a reactive part that can go and methylate the DNA. So that is consumed over in that segment. Now the other component follows the normal ADME process of redistribution and getting excreted from the body. So it's not that it stays over there for whole life. So it continues. This ADME process continues, resulting in the elimination of the metabolites from the body as well.
So are these residues or the metabolites are toxic to the normal cells as well when we talk about the toxicity, or not?
These metabolites, as of now, I may not be able to comment on that part because I have not looked into that aspect. But particularly this diazoniamine, which is the methyl diazoniamine, this is having toxicity because it can methylate, right? So it's a reactive component. So even if it goes to the normal cells, it is going to methylate the DNA over there as well. So,
I was wondering, you have just done some apoptosis studies. So did you check the apoptosis level in non-treated, nothing? These are the normal cells, normal physiology, and just treat with the metabolites. What would happen with the apoptosis level?
So we have not checked with the normal cells, the apoptosis part. But this is definitely going to cause because this is a mechanism by which it is going to affect. So if we are having methyl diazoniamine, if it is present, it can go and methylate the DNA. So if the DNA is methylated, then the cell has its own processes to repair. It can, because DNA repair mechanisms are also there, right? So if these repair mechanisms kicks in, so depending upon the status of the methylation, how much is happened, how much it has exposed, either the cell proceed towards apoptosis or it can recover.
Okay.
So that will depend upon the exposure.
Thank you so much, sir, for your comment.
So if we are having this methyl diazoniamine in the plasma, so that can lead to these toxicities which are related to this molecule, that hematological toxicities which temozolomide is having.
Okay. Thank you.
Thank you very much, sir. Now, one of the participants, Dr. Neeraj Verma, she want to ask one question, that temozolomide produces convulsions as a side effect. So whether this affect the animal studies or not?
As such, in our animal studies, we have not seen that observation of having the convulsions. So I may not be able to comment that, but definitely in any animal study, if such thing is happening, which is not acceptable, we need to work out on that part. So is this a journal thing which we need to see? But somehow in our studies, we have not observed the convulsions.
Okay, and one of the participants, Nitish Kumar, he said only 10% temozolomide reaches to brain. So is there any distribution study of remaining drug in other organ, or is drug showing secondary effect?
So basically, it has been shown in the literature that 10% is going to the brain. The remaining is basically metabolized in the systemic circulation itself, forming the MTIC and the diazoniamine. And this diazonium is basically causing the toxicities which are associated with it.
Okay. Thank you very much, sir. And can we mimic this model in zebrafish? Glioblastoma multiforme?
Yeah, I'm not sure about that, but definitely it could be looked into. Even a few days back, I was discussing with one of the researcher for having a zebrafish model for glioblastoma. It will be really interesting to see that.
Yeah. Okay. Okay.
So I have one more quick question. Then.
Yes, Maury.
So is there any information or the report that shows that the availability of this drug is different in male and female brain?
Male and female brain. That's an interesting question, but I have not come across for such kind of because what happens when we are talking about cancer therapeutics, right? The clinical studies are again done in the patient samples who are having tumor, right? and the objectives are very limited. As such, I'm not come across over this kind of a study where they are looking for the distribution part.
Yeah, because this is a major question nowadays in neurological disorders, because most of the drugs previously was considered as effective in both the sexes, but now come out with the conclusion that they might not be effective in one sex, maybe in males more than females, just because of the physiological difference in both the brains and physiology and metabolism and, you know, even a lot of things.
Yeah, no, definitely these do happen, right? But I've not seen any report as of now. Probably it may be there. I'm not sure about it.
Maybe I would like to like talk to you after this session, because I am just the guy who's working in the sex differences right now. Because I have observed a lot of things in brain and blood-brain barrier and brain region heterogeneity and astrocytic and glials and neuronal, even the inter-regional diversity. So,
Great.
So that is kind of the reason why most of the drugs nowadays not applicable to both the sexes, in males and females. And this is the precision drug era, I would say.
Okay, great. No, I would love to discuss more with you on this. Yeah, definitely.
Thank you.
Yeah.
Thank you very much. Now we have with us Dr. G.D.Gupta. Sir, you want to share your views?
Oh, so first of all, welcome Dr. Deepak Chitkara ji, and thank you very much for nice talk and very informative and excellent discussion. So ultimate goal and purpose of any types of the technical presentation, if the audience they can be involved and interact with the speakers, then it will be always beneficial. So obviously, because most of the researchers, scientists, they are presenting their own work. That is why they have the lot of information. Even at the end, they have also number of the difficulty they have the feel or found during the work. And in with this work, as you know that we are established here the Zebrafish lab. So always Dr. Arti sees the searching the opportunity, the field in which we can include also number of the models we have the established. And I know that the BITS Pilani pharmacy department doing the different types of the work, and most probably they have the minimum, they have the collaborative work like that. So I also request to Dr. Deepak ji, because I know that WITS is doing excellent job work. So if you have some of the collaborative idea and if you want to work with the ISF College of Pharmacy, our team, so I welcome you, sir. And from my side, I will send some of the proposal, because I know that if these types of the institute work together and the young mind over is the here, we have the good department of the pharmacology, zoetics, chemistry and ecology. So here is I don't say that we have the equivalent facilities, but we have the some of the good facilities in the pharmacology department also. So there is the number of the participants. So I request to all of the participants, because I am very happy. Dr. Mohit Kumarji is always involved in each and every session. So I feel that everybody, they can give their views. Sometimes if you are not asked the question, there is no problem. You can write down on the chat box what types of the field nowadays during this period, or you have learned something from this lecture or not, so that it will be work itself as a feedback, and it is a, you can say, one of the booster dose of the speaker also, because speaker always want what he contribute and delivered over here, what is the output and what is the person thinking about like that. Because nowadays what I have the feeling, if the speaker feel that I have the 1 hours and I have the only responsibility to talk and finish, the job is over, and I feel that I have the responsibility to listen 1 hours and finish the job over, then it will not come out any fruitful result. Because either international agency or national agency, if they are sponsoring some of the activity, the purpose behind this one, key number of the peoples, they will be the skilled and they will be learn some of the new technology, new concept, and they can be include in their life also. So thank you very much, and I again request to all of you, come out and you can discuss or you can share your views number of time I feel that you have the lot of courage and lot of efficiency or lot of, you can say, technology inside in your brain, but unable to express it. So this is the one of the difficulty of our country or some these types of the state. So if you want to the grow, you will discuss and you can the come out. Doesn't matter what you are the asking. If you can ask the very simple to simple question, it means you are involved with this talk like that. So thank you very much. Thanks to all. And if we can go through the on the screen 37 of the participants, and I have also one of the list over here. Near about the 80% of the participants, they are at that time present over there. They are continuously present in the all technical sessions. So this is the good thing, and congratulate to the organizer, because time to time we are the doing all of the activity. Thank you very much, Dr. Deepak ji. I have visited only the one time BITS Pilani Goa campus, and I have not received any opportunity to visit the BITS Pilani of the Rajasthan. But I am highly interested to visit this one. Not only professional, because I have never mentioned that I am scientist, but yes, I have mentioned why the focusly I am good administrator, good teacher, and good person like that, because I am not a scientist. Because I on this chair and in this room, how we can say I am a scientist? But yes, I want to contribute each and everything in the startup, in the innovation. What are you performing? Sometimes it will be the give the more. And today I have given one of the example during the one of the lecture. This is related to the one of the things bakery product, so that the life of the person is the growing. So number of time we are the doing the some of the small practical at our home with the food material, and how it will the useful for the diet complementary substance for the anemic patient. So this types of the thing most probably we are the trying. And we have also one of the YouTube, I have put one of the things. This is known as the medicated good park. During the lockdown period, I have prepared with the 18 substance we have added in the good park. And this is the, I feel that this is the good immunity booster like that. So these are the general things, and what I am listening, what I am thinking, what I am watching, and what is the coming surrounding throughout you, you can utilize, because government of India wants not to do a big research. If you have time, I want to quote our example here, because Dr. Deepak Chitkara ji, he is the belonging to the Rajasthan unit. One of the person, Dr. D.S. Mittal, he is in Lachu College of Pharmaceutical Sciences. He was the principal. Right now, he is the only just retired. And he served the institution 38 years as a teacher and 22 years as a working industry person. जेंटलमैन में ये बता देता चाहता हूं, उन्होंने एक भी एम फार्म नहीं, सॉरी, एक भी एमएससी नहीं कराई है, एक भी पीएचडी नहीं कराई है, और उनकी कंसल्टेंसी 1 महीने की, इफ आई एम नॉट रॉन्ग, more than 20 lakhs rupees per month. More than 20 lakhs rupees per month. I have received one video call after the 25 year, and he mentioned that, Dr. Gupta, you are doing a difficult research, but I am doing the simple research. And simple research always give the output. जैसे let us consider, डॉक्टर क्या होता है, पढ़ाई कठिन है, बट आफ्टर द डॉक्टरी काम सरल है. पेशेंट क्या आता है, प्रॉब्लम लेके आता है, प्रॉब्लम सॉल्व, यू कैन अर्न एनी अमाउंट ऑफ द मनी. तो वो कहते हैं हम प्रॉब्लम सॉल्व करते हैं, सर दिस इज द वुडन प्लेट्स, बट आफ्टर सम टाइम इट चेंज द कलर, नो प्रॉब्लम, आई फिक्स द कलर ऑफ द वुडन, you can pay the 5 lakhs rupees, because most of the material of the wooden, they are the export quality from Rajasthan. Sir, this is the arm, this is the mango tree wooden, but it have the yellow color after some time. Okay, I bleach it, give me the 5 lakhs rupees and you can earn so many things. I was the witness of this fellow, ki how they are the doing the work. He feels that I am not doing the original work, I am resolving the problem. He is the person fix the color of the bande sari, and 80 कंपनी के लिए वो आपको क्या देते हैं, कंसल्टेंसी देते हैं. डॉक्टर एस बी गोखले, वन ऑफ द पर्सन, डॉक्टर एस.वी. गोखले, जिनकी मोर देन मैं समझता हूँ 200 किताब होंगी, एक दिन मुझसे मुलाकात हो गई। बोले सुतली बम जानते हो? हमने यस। बोले आई एम गिविंग द कंसल्टेंसी टू सुतली बम। हमने ओ माय गॉड, फार्मेसी पर्सन, हाउ टू गिविंग द कंसल्टेंसी ऑफ सुतली बम? तो उन्होंने हमें बताया कि आई हैव प्रिपेयर वन मिक्सचर ऑफ द गॉन्स, डिफरेंट डिफरेंट गम्स, और जब मैंने पोटास को 1/4 कर दी और गम को रस्सी में डिप करके उसको लिपेट दिया, क्रैकिंग इज द 4 टाइम्स। यही तो बंदा चाहता है। अरे बम का मतलब आवाज। और he is the earning every year near about 10 lakh consultancy. So here is the number of the peoples. Then we are talking any types of the disease, any types of the project, any types of the thing, उसको economic mode पर जाके how to be simplify it. ये आपकी स्ट्रेंथ है, ये आपकी रिसर्च है, ये आपकी वैल्यू है, और यही मार्केट में बिकने वाली है। बिकॉज गवर्नमेंट ऑफ इंडिया वांट to produce the person as a self-innovator and startup. They don't want to the person, they are the searching the job and job. And every day in the newspaper, the thousands of the vacancy and lakhs of the people, they have the no any job role. So this types of the things is the new today. 1% establish the unit and provide the job role for the hundreds of the peoples like that. तो आज के रिसर्च के बाद, इस पैनल के बाद, इस इब्रो के बाद, आई फील दैट कि कम से कम एक इनोवेटर या स्टार्टअप जरूर चालू करे। देन आई फील दैट कि आवर सिस्टम और आवर इंस्टीट्यूट डू सम कंट्रीब्यूट फॉर द सोसाइटी। तो थैंक यू वेरी मच दीपक जी। ये सिंपल बात है, मैं इसलिए कहना चाहता था कि हम जागते रहो। एक आज बहुत अच्छा शब्द किसी ने कहा है कि रात में तो सपने सब देखते हैं, ठीक है, लेकिन सपना वो जो लोग देखते हैं, जो उसको साकार करने के लिए होमवर्क करते हैं, तो होमवर्क करना बहुत जरूरी है। सपने देख करने से अकेले कुछ भी नहीं हो सकता है। तो मेरा ये सोचना है, आइदर यू आर डूइंग द वर्क ऑन द पॉलीमर, नर्वस सिस्टम। आजकल टेस्टिंग की क्वालिटी बहुत अच्छी आ गई है, आजकल फैसिलिटीज बहुत आ गई हैं, सुपरफिशियल थिंग्स बहुत ज्यादा आ गई हैं, ड्रग कॉम्बिनेशन बहुत सारे आ गए। इसके बावजूद भी इफ आई एम नॉट रॉन्ग, अगर किसी के यहाँ प्लास्टर लगा हो, कभी देखा हो, एक लपेटते थे पट्टी और पानी से ऐसे ऐसे करते थे तो प्लास्टर ऑफ पेरिस हार्ड हो जाता था, वो आता ही नहीं है। दिस इज द डिसअपियर कंप्लीटली। टेक्नोलॉजी इज द न्यू एंड दे हैव नोन इट। और उसमें एक बड़ी प्रॉब्लम होती थी कंप्रेशन की, जिसमें रुई के साथ उसको आपको लगाना है, समटाइम इट विल बी द हाईली टाइट और बोन डिस्टर्ब हो जाती थी। ये डॉक्टर की एक क्वालिटी होती थी। आजकल तो मटेरियल बहुत सारे आ गए हैं, तो इन सभी चीजों पर हमारी हर रोज की चर्चा चल रही है। दैट इज वाई आई रिक्वेस्ट टू एवरीबडी, यू कैन इन्वॉल्व, यू कैन कंट्रीब्यूट, यू कैन शेयर योर व्यूज व्हेन एवर यू हैव द टाइम। और जब डॉक्टर आरती अलाउ करे, आज इनने अलाउ किया इसलिए मैं बोल रहा हूँ, क्योंकि अभी अगले लेक्चर में हो सकता 5 मिनट हो। सो व्हेन ही अलाउ टू मी, देन आई विल टेक द 5 मिनट्स। ले लेता हूँ, ले लेना चाहिए, ताकि मैं अपना शेयर की जिंदगी का मैंने जो किया है वो थोड़ा कंट्रीब्यूट कर सकूं। Thank you very much.
Thank you very much, sir, for your motivating words.
Yeah.
Yes, Dr. Deepak, sir.
Yeah, even I just want to add, I mean, sir, आपने काफी अच्छे points बताए हैं. It's really good. Even I want to tell the audience also that if you are doing something, you should look from the product perspective, that how it can reach to the market, not only just doing the research work. And in fact, over here, I have also started a company, a startup by the name Nanobreed Innovations Private Limited, that looks into the translational activities, particularly in the nano area, because there are a lot of research that is happening, lot of good results are being reported. If there are 1,000 publications, I mean, at least one product should come out of it, but that is not happening. So there is a big lacuna that needs to be filled. So if you can identify that at a very beginning, that what are those problems in the translation of a product, then you will be able to do your research, align your research so that it could be converted into a product, and maybe a startup, or maybe, you know, licensing it out. There are many opportunities. Yep.
So thank you very much, Dr. Deepak, sir. And you have discussed various important aspects of nanoscience and nanotechnology, and especially we would like to thank you for sharing your views and your research work with us. So thank you very much, sir.
एक मिनट में कंट्रीब्यूट करना चाहता हूँ, ओनली 1 मिनट।
यस सर, यस।
एक अभी हमने एक बाइट थी, आपको ये बताया, अभी मैंने एक 15 रुपए का बिस्किट्स खाया है। तो अभी डेलीशियस वाइट एक लेडीज यहाँ थी अभी से 30 मिनट्स बिफोर टू दिस वन, एंड ही स्टार्ट द बिजनेस विद द 2 kg ऑफ द मटेरियल. आज वो कहती हैं 2 क्विंटल ऑफ द मटेरियल एवरी डे सी इज द कंज्यूमिंग, एंड कॉस्ट ऑफ द वन इज दिस इज द 50 रुपीज. 180 रुपए का पैकेट. वो पैकेट भी दे गई है, हमें खिला भी गई है. ठीक है. तो और उनका नाम है डिलीशियस वाइट्स, जो कि बहुत जल्दी ऑनलाइन होने वाला है. एंड सिंपल ओनली द BSc लेडी, यानी कि देयर इज द लॉट ऑफ अपॉर्चुनिटी एवरीवेयर. X over to Arti ji.
Thank you very much, sir. Thank you very much. So we have our next speaker of the day. She's from Amity University, India. So Dr. Indu Singh, she is currently working as an assistant professor in the Center of Pharmaceutics, Amity University of Pharmacy, Noida, UP. She has also worked as an assistant professor in the Department of Pharmaceutical Sciences, Baba Saheb Bhimrao Ambedkar Central University of Lucknow. She has 6 years of post-PhD teaching experience. She possesses bachelor's of degree and master's of degree and doctorate degree in pharmaceutics. She has done her PhD in the area of nanomedicines from Naipur, Hyderabad. She has been a gold medalist in M.Pharm and B.Pharm. She was awarded with Sumitra Devi Vahi Memorial Award for Best Outgoing B.Pharm Final Year Student of the Year in 2008. She has been a recipient of Graduate Aptitude Test in Engineering Scholarship by Ministry of Human Resource Development (MHRD) and Fellowship from Ministry of Chemical and Fertilizers, Government of India, for pursuing M.Pharm and PhD respectively. She is a registered pharmacist with Pharmacy Council of India, Uttar Pradesh Pharmacy Council, Lucknow. She qualified Women Scientist C examination conducted by Department of Science and Tech, New Delhi, India in 2015. She was also awarded with Young Scientist International Travel Grant from Council of Scientific and Industrial Research, India, to attend 6th International Conference on Nanomaterial Research and Application, NanoCon 2014, at Czech Republic. She has several awards to her credit for oral and poster presentation in various national and international conferences. She has published more than 20 research papers in international journals of repute with good impact factor and is author of 6 book chapters and 1 book. She has also filed 2 patents. So we welcome you, ma'am. I think there is some technical error. I just confirm it from the speaker. Dr. Indu Singh.
Yeah,
Ma'am,
I'm so
kindly
sorry, Dr. Arti. I'm so sorry. I mean,
okay,
they, I
ma'am.
mean, there's some university things. I mean, they have blocked it from their end. So I had to contact the administrative
Okay.
person, and then I got it rectified. I'm so sorry.
No issue, ma'am. Are you able to present from your side?
Yeah, I'm able to do it.
Okay. You can continue, ma'am.
Welcome, Dr. Induji. No problem, because this is the technical problem. Sometimes it may take place.
Thank you, sir. Thank you so much for your time and patience. I mean, I was troubled. I tried contacting, and then I got to know that yes, they have blocked it from university site only.
Okay. No
Because
problem.
sometimes, yeah, I mean, the strength is very much here. I mean, the strength. I mean, the students also do that. So, I mean, that's why. I mean, due to some privacy things, they have blocked it. Google Meet. Yeah.
Okay.
Yes, ma'am. It is visible. You
Yeah.
can go to slideshow mode and you can start.
Yeah. Glioblastoma, which was previously known as glioblastoma multiforme, which is these are the tumors inside the cranium, the central spinal canal, and these are. I mean, we all know that the tumors are abnormal and uncontrolled cell division, usually the brain itself, but also sometimes they appear in the lymphatic tissues, blood vessels, cranial nerves, meninges, skull, pituitary glands, or pineal glands. The involved cells may be neurons or glial cells sometimes, astrocytes, oligodendrocytes, epididymal cells. And brain tumors, definitely, whenever we talk about cancer, they can be metastatic, they can be benign. So, yeah. Moving on to the next slide. We have certain data, the statistical data. This is the statistical data which I got from the WHO. According to they have standardized the incidence rate for brain and central nervous system tumors.
Sorry for the interruption, ma'am. Slide is not moving. Slide is
It
not
is not
moving.
moving.
Yeah.
Okay, wait. Let me stop and share it again. Let's type it. I
You
mean,
can
whenever
continue.
I'm doing, yeah, as soon as I'm doing the slideshow, it is gone. But is it
No need,
moving?
ma'am. You can continue in this mode also.
Okay.
Yeah, it is moving. Yeah.
So I mean, I'll zoom it, and then you'll be able to see that. Yeah. So this is the incidence rates of the glioma, which is yeah, the brain tumors. I mean, WHO standardized the incidence rates. I mean, I got it from the WHO website. So you can see it here. The incidences as I mean, this is for the blue ones are indicating the males, and the magenta ones are for females. So you can compare it here. This from southern Europe, the incidences are more as compared to the, I mean, for what do you say? We lie there in the Asian region, right? Asian. Yeah. So Eastern Asia, it is showing the incidences rate is around 4 in yeah, 4% and 3.6 in females. So similarly here, glioblastoma multiforme, what is it? This is the most common form of the brain tumor that accounts for about 46% of the intracranial tumor with a risk of 30 to 100 per millions. Second cause, this is basically second cause of cancer death in adults less than the 35 years of age. I mean, this is it happens basically in the young age population. So average survival expectancy is around 40.6 months, and overall 5-year survival rate for glioma is around 9.8%. So we basically target, our group is working on the designing of the surface functionalized nanocarriers to target to the brain cancer, brain tumor, that is the glioma. So what hurdles are we going to find in the therapy? That is the poor permeability. Whenever you talk about any targeting to blood brain, I mean, the blood thing or the brain thing, the brain tumor, I mean, the most important hurdle you come across the blood brain barrier. You need to bypass that to internalize your uh, molecule or your system, the functionalized system. So yeah, we have specific cellular barriers that can be overcome by the EPR effect or the overexpressed receptors. By making use of these effects, we can uh, target our drug to the brain, the system to the brain. Lack of specificity and poor distribution, that can be taken into consideration whenever we are using certain ligand or bioconjugation thing. Then we can overcome this specificity and poor distribution. Multi-drug resistance and drug efflux, that can be overcome by the substrate of PGP. I mean, multi-drug resistance is a very common thing. The uh, permeation glycoprotein is one thing which, whenever we used to target something to drug, this comes into picture a lot. So what strategies do we have for targeting to blood-brain barrier? Definitely we come across three things basically. First is your absorptive mediated transcytosis, which is nothing but your clathrin mediated endocytosis, right? Like the substrate or the metabolites internalize into the brain. I mean, that is similar to that. Transporter mediated transcytosis is something I mean, we all know the some transporters or glucose transporters are always present on the brain. So making use of that channel, we transport our, I mean, using those transporter as some ligand, we can transport our molecule or we can transport our drug or system to the brain. Receptor mediated transcytosis, again, we all know that some receptors are expressed on the brain, or I mean, they may get overexpressed whenever, I mean, in case of certain diseases. As we all know that the in brain tumors, certain receptors get overexpressed, like lactoferrins are there, transferrins are there. Right. So making use of those receptors, we can target our drug or we can target our system to the brain. So what are receptors? Basically, to introduce, a receptor is a protein molecule usually found embedded within the plasma membrane surface of a cell that receives chemical signals from outside the cell. So now how we are going to use that? Why receptors are so important? Yes, these are receptors have certain importance because they, by making the use of those receptors, we, I mean, if we are going to bind certain receptors, bind certain proteins onto your molecule, onto your system which you are going to target, it will help in the internalization of that particular system to that particular disease or to that particular area. Like we all know, certain receptors which are overexpressed on the brain are folic acid receptors, adenosine receptors, sigma receptors, transferrin receptors, lactoferrin receptors, glycoprotein receptors, etc. So appropriate ligand can be chosen to target these receptors. Yes, I mean, based upon your knowledge and understanding of the receptors, you can choose any of the ligands to target to certain disease. Yeah. So now there are certain and several ligands are there which can be used as your target molecule to target, like proteins are there or small molecule ligands are there. Now the question comes, how are you going to select among the proteins or small molecule, which ligand is going to be of your choice, right? So how you're going to decide upon that? We have certain parameters on which we are going to look into, and we are going to decide upon which ligand is better in what terms, right? So let us see. Ligands, we have peptides as well as we have small molecules. So why peptides and why small molecules? Who is going to be better ligand, right? So we have characterized certain differences, among based upon that we can choose our respective ligand. So for peptides, definitely we know that, these are long chain proteins, right? So they'll be large in size, and they will have a certain structural complexity, and they'll be unstable. Also, they are going to be costly. If you're going to use it, the cost effectiveness will not be there. So and choosing the ligands, we'll have to be a little careful. A difficulty in conjugation. Definitely, if you're going to conjugate the chemistry, conjugation chemistry, due to its bigger size, the conjugation will be difficult in the system. So cumbersome characterization, definitely due to its large surface area, large number, long chain, it is going to be difficult. But since it has a defined structure, the targeting based upon the targeting using these peptides is going to be better. Whereas we have the small molecules, like small size molecules, these can be simple, these have simple defined structure, they are stable, and definitely due to the size, cheap, the conjugation is easy and so we are going to get a better conjugation efficiency, better characterization, but the targeting potential is somehow compromised. So receptors and transporter which are overexpressed on blood-brain barrier, there are certain receptors like transferrin, melanotransferrin, lactoferrins, sigma, leptin, LRPs, glucose transporters, folate, LDL, interleukin, integrin, and insulin. Let us see. This is the most common barrier whenever we talk about any brain disorder. Whenever we are going to target some drug to any brain disorder or brain disease, we have to bypass, we have to always look into the details of blood-brain barrier. What is blood-brain barrier? It is a dynamic barrier protecting the brain against invading organisms and unwanted substances. It is also the most important barrier impeding drug transport in the brain by the blood circulation. It has very particular characteristics, lack of fenestration, and with very few pinocytic vesicles. The presence of tight junctions between adjacent endothelial cells formed by an intricate complex of transmembrane proteins, expression of various transporters, lack of lymphatic drainage. This system is very essential for normal function of CNS. It regulates passage of molecules in and out of brain to maintain neural environment. So let us see with, we have targeted the brain tumor through using the small molecule as well as the peptide also. So this one is the small molecule glucose 1 transporter through which we are going to target the disease. Yeah. So para aminophenyl alpha D manopyranose, which we have shortly named it as MAN. So this is one important glucose transporter. This mediates its transport across the blood-brain barrier. The structure is mentioned here only. It has one free amino group, right? is easily attached to anchor groups available on the nanoparticles surface. This is a small molecule ligand. It is a sugar derivative able to traverse the blood-brain barrier. Now coming to the peptide which we used in our research, this is lactoferrin, which is a polypeptide chain of around 690 amino acids, which belongs to the transferrin family. It is a mammalian caninic iron binding glycoprotein. The we all know, I already demonstrated, I already told you that LFR exists on the blood brain barrier, and in some and they are overexpressed on the glioma cell surface. Whenever someone is having they gets overexpressed in the disease case, in the brain tumor cases. So the system of our choice was solid lipid nanoparticle. Why solid lipid nanoparticles? Because as we all know that being lipidic in nature, the lipophilic drug can be easily entrapped into the cavity, or you can say it has better physical stability, right? So different lipids can be explored for changing the properties of the system. We can use different type of lipids, whether it is acidic, whether it is containing acidic group, amino group, whatever it is. So the ease, due to its large surface area, the surface modification is easier. They can be in submicron size, around 50 to 1000 nanometer particle size. They are made up of lipids, right? So lipid aids in permeation from various barriers, including the blood-brain barrier. So how did we prepare the SLNs? How did we conjugate the ligands over the surface? Let us discuss that. Preparation of drug-loaded SLNs. We use the simple solvent emulsification and evaporation method. What we did, we prepared one aqueous phase and organic phase. In our aqueous phase, we kept the simple the 280, and in organic phase, we kept the what is that ethanol, and your drug of our choice was docetaxel, right? So it has been explored with various systems. So conjugate, how did we how did we got the conjugation? The conjugation, it was achieved using simple carbonamide chemistry, EDC NHS chemistry. So we, since the SLNs, the excipients used in our SLN was stearic acid. So as we all know that stearic acid has a free carboxylic group in it, and we already discussed the uh, mannose conjugate, the mannose ligand which we used had one NH2 group in that. So making use of that chemistry, we conjugated the ligand over the SLNs using the EDC NHS chemistry. So this is how the conjugation was achieved. And for the conjugation efficiency, we calculated it through one method. I'll elaborate on that too. So how did we characterize the particle size? It was characterized using the zeta sizer, yeah, DLF size, dynamic light scattering. The size, particle size was around, for the conjugated, it was around 100 nanometers, and unconjugated, it was around 85 nanometers. DSC studies were done to check how the drug, the amorphous nature, whether it is going into the amorphous form or not. So the DSC thermograms of DTX, PM, and mannocellin we have already given you here. It is showing that mannocellin is converting into amorphous form or the disorder crystalline phase. FTIR, yeah, this is very important to show the conjugation. How the conjugation was achieved, since we know that the free acidic group was there in the SLN, and the NH2 group of the ligand was going to combine. So we are seeing that the CO stretching, CO acid stretching, which is around 1723, is not appearing in the mannose SLN1. Right? Can you all see the image? Can you all confirm whether you can see it?
Yes, ma'am.
Yeah.
It is visible.
Yeah. Okay. So the acid stretching is not there in the mannose SLN FTI spectra. It is clearly. That is one evidence that, yeah, the conjugation has been achieved. Also, a peak at around 1640. We all know that CO amide stretch comes around 1640, right? So amide stretch, this peak is of amide stretch, which is showing that yes, one conjugation between COH and NH2 has been done. Similarly, another confirmatory peak was NH bend around, it was around 1552, which is showing that the conjugation is done. This is how the FTI was useful in showing the conjugation. Now, coming to the in vitro studies, we performed the in vitro studies, release studies on the drug suspension as well as the unconjugated and conjugated SLN. So, USLN are the unconjugated SLN and MAN-SLN is the mannose conjugated SLN. So, I mean, we performed the studies in PBS 7.4. Yeah. So, from drug suspension, more than 80% of the DTX was released in 12 hours, whereas the unconjugated SLNs exhibited approximately 33% release of DTX. DTX is nothing but your docetaxel in 24 hours, whereas the conjugated SLNs displayed about 25% of DTX release into. So it is giving your sustained release, right? Formulation stability was checked. I mean, various parameters was checked. The particle size, zeta potential, and entrapment efficiency. When we go into this, we see that the particle size of the unconjugated SLNs were around 78, which which are going to be bigger in case of man SLNs. I mean, the conjugated ones, the size is going to be increased. Definitely, I mean, if you're conjugating something on the system surface, the size is going to be ultimately increased, right? Zeta potential was, I mean, due to the acidic nature of the steric acid, the zeta potential was somewhere moving to the negative side, and which got better when we attached the ligand onto the surface. Similarly, entrapment efficiency got decreased after conjugation, because we used certain chemistry, the coupling chemistry, through which the drug, it is possibility is there that the drug may come out during the reaction. Cytotoxicity in U87 mg cells. Yeah, we did the cytotoxicity studies also to check how they are going to be, how the formulation is doing in the in vitro, in the cells, right? The MTT assay was done. So, I mean, the result is showing the first figure is showing you the inhibition, percent inhibition on U87 mg cell, which is, I mean, which is treated with DTX formulation for 48 hours. A is your no your U8 for normal cells. B is docetaxel solution. C the unconjugated SLNs, and the man conjugated SLNs. Cytotoxic images obtained after sacrificing cells with study formulation. The concentration is around 1000 nanogram per ml DTX concentration. Reduction in cell density was observed after treating cells with CSLN. You can see it here also in the table. IC50 value is going to be decreased in case of the conjugated SLN ones. Cell uptake studies. Yeah, I mean, we have shown the figures, some pictures, phase contrast images, and fluorescent images showing uptake of different FITC tagged drug-free lipidic formulations after 2 hours. And the data suggested that FITC tags conjugated SLNs have shown more uptake compared to that of the unconjugated one. You can see it in the table 2. The uptake was more in case of conjugated SLN. It was around 17.9 plus minus 0.29%. We also did the pharmacokinetic studies. Yeah, we performed the pharmacokinetic study, and some of the pharmacokinetic parameters, these were done on the female Swiss albuminides. And the pharmacokinetic parameters which we evaluated are listed here. Plasma Cmax, I mean, we compared the formulation, the unconjugated and conjugated one with the marketed one, Taxotere, and we found that the Cmax was, improving, right? And plasma AUC was around 70.43 plus minus 4 in case of the conjugated one. Half-life got was around 2 point, which is not, I mean, that data is not much significant, but yeah, it has and the half-life got increased somehow. Mean residence time, similarly, it improved in case of the conjugated ones, conjugated formulation. Brain distribution studies, we did the brain distribution studies too. Details concentration was found in brain. We have represented it through the pictorial representation. You can see it here. Brain concentration of DTX was elevated significantly with lipidic nanoparticle formulation at 1 hour. No significant difference in concentration of DTX was found for taxotere and mannocellin. DTX concentration increased considerably for mannocellin compared to that of unconjugated one at every time point. You can see it here, whereas taxotere was not detected. You can see these stars only, right? The taxotere is not there after 1 hour. So similarly, and that work was about the small molecule ligand which we joined on the SLN surface. Now coming to the another ligand which we use, the peptide one, lactoferrin. The same chemistry we used it here, the same method we used, solvent emulsification and evaporation method. By using the free NH2 group in the lactoferrin, we conjugated through the EDS-NHS chemistry, and we got the conjugation which was confirmed by the Bradford assay. The conjugation efficiency was estimated around 71%. Right. Now, how did we confirm the conjugation? The surface morphology was done using the atomic force microscopy. As we all see that, yeah, the 3D structure, the irregular flattened and 3D images of SLN, and the first one is for SLN and second one is for CSLN. So I mean, morphological changes and increase in the height of the height is going to be increased. As you can see it here, in case of SLN A image, the height is around 13.51 nanometer, which got increased in case of your conjugated after conjugation, which is around 38.50 nanometers, which is a clear indication that the yeah, so the attachment is there, the ligand has been conjugated. The Now again, the FTIR spectroscopy was done to confirm the linkage, the ligand conjugation through the amide linkage. Exactly, you can see it here, the again the CO stretch, the amide stretch, and the NH bend, which we see, which we observed in we observe in amide peak, is there clearly visible, which is a clear indication of the conjugation between the free NH2 group of lactoferrin and free group of the stearic acid, which is there, which is present there as the lipid in the SLN. Crystallinity studies were done using the XRD. XRD, I mean, you can see it here. DTX has several crystalline peaks, I mean, which after conjugation, I mean, which after preparation of the SLN, they got the peaks are distorted, or the peaks intensities are getting reduced. So this is a clear indication that the drug is going into the emulsiform. The drug is the crystalline nature is going to be reduced. DSC, again, we did the DSC studies to check. Similarly, we found out that in the C curve, the SLN conjugated thermogram, we clearly show, I mean, it is showing the conversion of crystalline DTX into amorphous disordered crystalline phase. In vitro drug release studies again were done to, were performed on all the I mean, SLNs, the unconjugated one, the conjugated ones, and the drug suspension. So we found out that the drug exhibited around 80%, more than 80% in 12 hours, whereas the drug from the SLNs, it was from SLN it was around 33% and 23% in conjugated ones. Colloidal stability studies were done to check the stability of the drug in the colloidal particle and the colloidal nature. Colloidal stability of LF conjugated solid lipid nanoparticles in normal saline and serum were done. Here we have given you data indicating trivial increase in particle size. Hence, particles are stable in serum and normal saline. Cytotoxicity in U87 mg, again we did the MTT assay in the U87 mg glioma cells to check upon the IC50, I mean, how it is reducing. We have given you one IC50 table here. I mean, it was around 0.857 for the pure DTX, and it has got significantly reduced in conjugated SLN1s. Ligand receptor binding assay was done to check the mechanism. I mean, IC50 obtained after U87 mg cell with different tested formulations, and comparison of these were done with the competitive ligand receptor binding assay. The IC50 value increased due to saturation of LFR with free LF. Cell uptake studies again were done to check the uptake in U87 mg cell lines. Yeah, graphical representation is always also there. I mean, you can see the cell uptake is much better if we compare the SLNs with the conjugated one. Four-fold increase was there. So apoptosis वैसे an exon 5 binding assay by mu stem cell analyzer after 24-hour treatment with DTX and lipidic nanoparticle formulations was done in U87 mg cell lines. Results explain that the conjugated formulations showed better apoptosis than the other tested formulations. So this work was done with our done by our pharmacology fellows, pharmacology colleagues here. Pharmacokinetics we did in the again same female Swiss albino mice. I mean, we are still looking for to check the uptake, to check the activity of the formulation in a tumor model. We haven't done the study, but we are looking into that. I mean, we are trying to collaborate for some for the study. Right, since we are lacking this facility, developing the cancer, developing the tumor, and so that is how we are trying to collaborate with some other to check upon the. This work is already been reported. This is already been published in the International Journal of Pharmaceutics and your Journal of Drug Targeting. So we are trying, we are looking for collaborations and to further explore for the study in models, tumor models. Right. So brain distribution studies were done, clearly showing that after 1 hour the taxotere was not there, but the SLNs were still there. They were active. Yeah, since we all know that lactoferrin is expressed in various tissues also, and apart from the brain, it is also present in liver, kidney, heart, spleen, and lungs. So we did the distribution studies in other tissues also. So we have done the comparative analysis for taxotere, the marketed formulation, SLNs, and the conjugated ones. So this was the study. We found that we have already demonstrated it through the graphical illustration of DTX concentration, which is found in unearthed tissues after different time intervals for different DTX formulations. Yeah, it was increased significantly with SLN and CSLN compared with the marketed formulation. So conclusions of my work was that what we found is that lipidic carriers are always a better choice for delivering certain systems to brain. You are going to improve the targeting efficiency due to its lipidic nature, physical stability, and large surface area through which you can conjugate a ligand over it. Right. Moreover, they are biocompatible and offer high stability and drug payloads. Although lipidic carriers can better traverse across the blood-brain barrier, but to improve their targeting efficiency, we conjugate a small molecule ligand as well as one peptide ligand, which has specific affinity to the glucose transporter 1 receptors and LF receptors. Was first time conjugated to SLN present one and evaluated for its efficacy through in vitro and in vivo studies. Based on the results from present, it can be concluded that formulating lipid nanoparticulate system can increase the efficacy of drug, and conjugating cell-specific ligand like the amino paraminoside and MAN and LF further can augment the site-specific delivery of the drug to brain. This is what we did. I'm open to question answers now. Thank you so much.
So thank you very much, ma'am, for your wonderful talk. Now the session is open for discussion.
May I?
Yeah.
Yeah, please.
Yeah, thank you so much, Dr. Indu, for this wonderful talk and sharing your research. So I have just few basic questions
Yeah.
about your work.
Yeah, please.
So first of all, what drug did you use to
Docetaxel. Docetaxel.
Docetaxel. Okay. So because you talked about something that alpha aminophenyl alpha D monopyrandoxide and GLUT1 receptor. So is
Right,
this,
right.
what is that? Can you please explain? So because I am kind of interested in GLUT1 receptors right now.
GLUT1 transporters are overexpressed on the brain, right? So we found, through certain literature search, we found that the receptors got overexpressed in case of brain tumors. So since the GLUT transporters are already there on the brain, so this ligand, by making use of this ligand, we conjugated the ligand on the surface of SLNs to increase the targeting efficiency, which is, this can be transported through glucose transporters, which are there on the blood-brain, brain tumor as well as blood-brain barrier. So to improve the targeting efficiency, we are doing this work.
So is there any chance we can modify this drug like this so that you can tag the glucose with this drug? Is it possible? Or 2DG, for example?
Yeah, definitely. You can explore that. Data is there. Various literatures are already reported that glucose can be transported. We all know that glucose are overexpressed. That the glucose receptors are overexpressed in case of cancer cells. That was the basis of our study. Yeah.
Because cells need
There are a lot
glucose
of
to
scope.
survive.
There is a lot of scope in this area. Yeah.
Okay, my second question is about the cell lines that you have used in your study. It says U87mg, right?
MG cell, right? Glioma cell lines.
Glioma cells derived from male patients.
Yeah.
But for your in vivo study, I think you have used the female mice. Am I right?
Due to some, definitely we use that because we had the availability of those things only. We were in hurry. We had to submit the data, submit the report to the DST thing. So we tried that. That's what we didn't do the tumor model study. We are still looking for that. We'll further explore the efficiency.
Okay. So you are also looking to use cell lines derived from females and
yeah,
in
definitely.
vivo males.
Right.
So we can say that this drug and nanoparticles are applicable to both the males and females equally and equally efficient to both of them.
Definitely.
Okay. Thank you so much.
Thank you.
That's it.
Thank you very much, ma'am. Any other participant? Okay, fine. Dr. G.D.Gupta, sir, would you like to contribute? Okay, it seems he might be busy. So, okay, thank you very much, ma'am, for sharing your research experience with us. It was a wonderful experience.
I'm extremely sorry, Dr. Arti, and all the. I'm extremely sorry. I'm saying sorry to all the participants. They waited a lot. I had certain network issue. I'm really sorry for that.
But finally, you make it, and
I made it.
It's wonderful.
Somehow.
Yeah. Thank you very much for your informative session. We really enjoyed it, and hopefully it would be beneficial for those who want to work in this particular research area.
Definitely. We are always open to collaborate, right?
Yeah. Thank you very much.
Thank you, Dr. Arti. Thank you, Dr. Arti, Dr. Charan, Dr. G.D.Gupta, sir.